Alright, hello biology, this is Mr. B, and we're continuing on today with section 3, except now this is part 2. So we're chapter 7, part 2, or chapter 7, section 3, part 2, and yesterday we talked about how we have cell membranes and cell walls that surround the cell, and now we're going to talk about how things can pass through that cell membrane. So the first term we're going to need to know today is diffusion. And diffusion is defined as the movement of particles from one area to another. This movement usually occurs by particles traveling from an area of high concentration to an area of low concentration. So what that means is it's just traveling to an area. Let's say we have 10 people on one side of the room and we have 5 people on the other side of the room. Diffusion states that the side of the room that has 10 people on it, maybe 3 people are going to move over to the other side until we have equal numbers. So it's going to travel from an area where there's a lot to an area where there's not that much. And diffusion in terms of cells, every cell is going to live in a liquid environment, and the cell membrane regulates the movement of dissolved molecules from the liquid on one side of the membrane to the liquid on the other side. So that's how diffusion occurs in cells. It's because it's going to be more in terms of liquids and things like that, because cells are always living in a liquid environment. Like, again, inside of our body is just an entirely liquid environment. And so when we look at when we look at diffusion, we have to look at something called concentration. So I said area of high concentration for an area of low concentration. So you might not know what that means. So concentration, how we measure it is we look at a solution, and a solution is just a mixture of 2 or more substances. And the substances dissolved in the solution are called the solutes. So when we think of, we have water and salt mixed together, that's a solution. We have 2 separate things mixed together. And then when we think of a solute, the thing that is dissolved would be the salt in that situation. And concentration is measured when we take solute in a given mass of a solution, or mass over volume. So when we think of concentration, that's what we're going to think of, the amount of dissolved particle that's inside of the solution. And so we're going to look at what happens exactly when during cell diffusion. So particles in a solution, again, this is a little bit review, or going over again, tend to move from an area where they are more concentrated to an area where they are less concentrated, and this process is called diffusion. And when the concentration of the solute is the same throughout the system has reached equilibrium. So we're going to look at some pictures, and it's going to make a little bit more sense here. So here we have a picture, and we're going to break down each set of here. So just take a look at that. See the solute here, only on this side, then it travels to that side, so that's the diffusion occurring right here. The particles traveling from an area of high to an area of low, and then we have equilibrium where they're traveling back and forth. So breaking it down step by step. So there's a higher concentration of solute on one side of the membrane as compared to the other side of the membrane. As you can see, all these little dots are the solute. There's a bunch over here and none over here. So our next step is solute particles move from the side of the membrane with a higher concentration of solute to the other side of the membrane with a lower concentration of solute. And the solute particles would continue to diffuse across the membrane until equilibrium is reached. So as you can see, they're going back and forth here, or not going back and forth, only going one way. And then our next step, there we go, there it is. Okay, and then when equilibrium is reached, solute particles continue to diffuse across the membrane in both directions. So you can see the arrows here are pointing in both directions, to whereas before it was only pointing in one direction. We can go back up. Okay, yeah, so see, arrows only pointing this way, and now we have going both ways. And then there it is again, just same picture, same thing, just again, more review so you can see what it looks like. And then diffusion depends upon random particle movements, therefore substances diffuse across membranes without requiring the cell to use energy. So this is going to be important. So the cell does not have to use energy, so no energy is being used by the cell when this occurs. And so something similar to diffusion is called osmosis. And so osmosis is the diffusion of water through a selectively permeable membrane. We'll talk about what that means in class, but if you don't know, write that down as one of your questions. So osmosis is a diffusion of water through a selectively permeable membrane. So diffusion, we had particles up here, let's see, go back, we had particles here, and when we talk about osmosis, these particles would just be water. And a little breakdown of what that looks like. Again, you can see there is less concentrated water on this side, or sorry, more concentrated water on this side, and this side has more sugar, and as you can see, the water is going to flow, so the water concentrations equal out. So that is what osmosis is. It's the same thing, except it's just water moving. You can see water tends to diffuse from a highly concentrated region to a less concentrated region, and if you compare two solutions, the more concentrated solution is hypertonic. So think of, you guys are always hyper in class. It means you're talking all the time. That means you're kind of going above and beyond. So the more concentrated is hyper, and the more dilute solution is hypotonic. So we'll try to go over these two again. But hypertonic means it is more concentrated. Hypotonic means it is less concentrated. And then when concentrations of solutions are the same on both sides, then we call it isotonic. So we have hyper means more concentrated, hypo less concentrated, iso equals the same. And then this osmosis just causes pressure on, again, on the hypertonic side of the membrane, so there is going to be a little bit of pressure caused by this water. But the cell is filled with salt, sugars, and proteins and other molecules, and it will almost always be hypertonic to fresh water. And if so, the osmotic pressure should produce a net movement of water into the cell. As a result, the volume of the cell will increase until the cell becomes swollen or bursts. That would be kind of in a bad situation, so we usually have things to control that. So that is all I have for today. Let me know if you have any questions.